Welcome to amplicon sequencing library preparation tutorial at Department of Food Science. Before we grab the pipette, there is a minimum amount of bureaucracy that we need to handle. Our protocol allows for simultaneous analysis of over 400 samples. So before you start preparing your library, you need to carefully select the primer set that you're going to use to mark your samples, and make sure that they don't overlap with other colleagues joining the run. So here is an example how to fill in such protocol. So for example, I'm going to sequence 66 samples and I have marked with green color all the barcodes that I'm going to use to mark my samples. Then I'm filling information regarding the sub library 01, so the next user will not use the same barcodes. It is very important to include this information on the tube where your pooled library will end up in. In this example, our run is called NextSec008, so we know that your sample is going to be sequenced on NextSec, not on MySec, for example, and that your sub-library is called 001, so based on the protocol that you fill in, we can find your name and we know which barcodes you have used. Plus, you can include the date that the library have been prepared. Okay, let's get started. The most important thing is to have your sample sheet prepared before you start assembling second PCR. If you have any sample missing, you don't have a full plate, you should prepare the tip box that follow exactly the same pattern as your PCR1 plate. You're going to need first box to transfer forward primers that will go to direction from the left to the right, and then the second tip box with exactly the same pattern to transfer your reverse primer that will follow from top to the bottom, and the third tip box to transfer your PCR1 product. We're going to need a sterile Eppendorf tube for our master mix, the fusion master mix containing everything we need to perform second PCR, and a bit of milky water that we can use only once to avoid contamination. Our master mix loaded. Now it's very crucial to check the order of the adapters, and you need to write them down on your protocol, because even if you swap two, then you will never know. So at this point, before we add, it's important to make sure what is the order, and if this is A, this is 7. Even if you swap, it's okay to swap, but you need to make a note. And then these cups are only single use, so all the time, every time you open them, they go to trash. And the reason for that is that these are all unique primers. So every primer has slightly unique sequence, And if you put, if you want to reuse them and you put them into another one, we will cross-contaminate them. And if you cross-contaminate them, it's also something we would never know, because there's no way to check if you cross-contaminate stuff, because then we pull them. So this is why it's just important to for the precautious reason. So now we'll be going this direction. Let's take a multi-channel pipette. This one needs to be set to pipette and blow. You can press R to calibrate it. And now the arrow up shows you that the pipette will be taking the liquid out. And then, so if you press, it goes up. Now it goes down to blow, and there is second blow. So you need to lift it up. I'll show you in a second. Like this. So, and that's why it's important to have the same pattern. So you make sure you have it. You go down to the tips. You press one up. Then it's important just to check if you have everywhere the liquid in. Can you see it? Mm-hmm. That is everywhere. Yeah. 2. Then you go to the first row. Release it and go a little higher to touch the well, and release it again. And now they dry. Then you discard them. Yeah. And now we know that we already loaded the first row. So now we go to the row 2, the same trick. Up, we check. I see that it's everywhere. And row 2. Out. Number 3. Tip box prepare, and we're gonna go in this direction, right? So it's the same pattern, and we go this direction. And in case you see that it's, one is missing, now it's more or less okay. But if it's missing, you can put it back. You can change the tip if it's necessary, or correct the tips, and then you go back again. And now actually it's really, this one is missing, right? Yeah. Okay. So I just put it back. You can correct the tip a bit. So we have added the last one. So now what you do is quite important, but before we open the PCR product, we need to close the primers. If we don't do it, you risk that the PCR product will end up in the primers, and that your colleagues will start amplifying your PCR product in the next reaction. So it's very important just to close them now. So you put the fresh caps. So now we have this one closed. this one ready. Now we just transfer with the same tips pattern the DNA. And the way I will go this direction, here, here, and then they will follow the same pattern. And when you open, you have to be very careful. So, because it's a PCR product, we don't want to make it too messy and contaminate everything. So you need to hold firmly to the round and support so it don't explode. This one also contains the PCR product now as a vapor, so not to avoid contamination, you can cross fold it like this and throw it, and then we're gonna use the new one to close it back. This goes now to the thermocycler, but the cooling block needs to be back here. This belongs to this lab only. And then make sure that each time you clean after yourself, and then you make the place as clean as possible. This goes back to the freezer. This is trash. Yes, and the primers go back to the freezer as well. Pre-book, right? But you need to yeah, just put your name on it and pre-book. So you just go to programs, and I guess it's on my name. Under Lucas there is a second PCR Nextera. Open. And actually it's always very good to check with your protocol if it's the same, if someone else's didn't make a change, and save it. So we can say we have 98 for 1 minute right here, and then the cycling of 13 cycles, 98, 55 and 72, 10 seconds 2020, which fit our protocol. So we can press start, tell them that it's 25 microliters. Okay, now it takes approximately 20 minutes. Everything clean. The first PCR, the best way to store it actually is to make sure that this is the seal is properly closed, and to avoid evaporation it's better to put it upside down, put it in the box. On top of the box we make a proper labeling, thank you, that it's a KK Maria. For longer period, and probably you will store for longer in the first and second PCR plates. So there is a special designator drawer for that. You can put it here, and after the sequencing is done, you can remove it. Or there is a project in progress, so we can put it here for a few days. So in case we see that there is a first or second PCR needs to be repeated, it can be stored here. DNA is very stable, and we put it upside down, so we will prevent evaporation. We leave it here, and if we see that the second PCR worked very well, we can put it to the freezer. And the same with the other stuff. So this goes back to the freezer. Water we can use single use to prevent contamination, so we trash it, while these guys goes back to the fridge. Indication that it was used today, so that's why we know that someone has freeze thawed it already once.
So we have 3, 4. So whenever we have a master mix for fusion that we use not all of it but half of it, we can after first opening we can put the little dot on it. So then the next user knows that this is this polymerase was opened once and it should use it first. So this way we don't freeze thaw the same polymerase many times. So that will be the first to thaw now.